All right. So now we'll be saying that after secretion from follicular cells, what happens to thyroid hormones. Now listen with all your attention. So suppose this is one follicular cell. Okay. Suppose this is one follicular cell. Okay. Now suppose this is your blood vessel. This is your blood vessel. Now listen with all your attention. So from here T4, T3 enters bloodstream by the help of monocarboxylate transporter, right? MCT8, right? MCT8 type. Okay. Now listen with all your attention. After entering the bloodstream, what happens is this. After entering the bloodstream, the T4 and T3 now binds with a protein which is produced by the liver. Okay. Now listen. What happens is this. Liver secretes a protein into the bloodstream. Which is called thyroid binding globulin. Okay. Now listen with all your attention. Now actually thyroid binding globulin is one of the protein actually secreted by liver into the blood cell for binding T4 and T3. Now listen. There are actually two other proteins. There are in total 3 proteins which actually binds with T4 and T3. Number 2 is called, that is albumin. Okay, albumin also binds with T4 and T3. Clear? Another protein which does the same job. This is called transthyretin. Okay, transthyretin. Okay, so there are actually 3 proteins. Okay, there are 3 proteins which actually comes into the blood from liver, which actually helps in binding of T4 and T3. Okay, suppose this is your thyroid binding globulin, or TBG. Okay, suppose this is your TBG. Okay, thyroid binding globulin. Now listen with all your attention. Now important point to understand is this, that T4 have higher affinity. T4 have higher affinity for thyroid binding globulin. Okay, so affinity towards TBG is T4. Okay, so that means quickly T3 will now bind here. T4 will now bind here, right? Okay. Now listen. Almost 99.98% of T4 binds with TBG, or thyroid binding globulin. 99.98% binds with TBG. Okay, and almost 99.5% binds with 99.5% T3 binds with TBG. Okay, therefore it has got higher affinity. Now the binding with TBG for T4 is very important. Why? Because actually T4 T4 acts as a pull for T3, you know. T4 acts as a pull for T3, okay. That means in your cell, suppose this is your cell, okay. So this is your cell. If T4 enters your cell, okay, then it will be converted into T3. Clear? It will be converted into T3. Clear? But if you don't have T4, then from where you will convert this T3? Okay, that means T4 is actually the reservoir or pool of T3 in disguise. Clear? For your, you know, body. Okay. Now we will see that what will happen if this T4 or thyroid hormones will reach a cell, okay. Clear? Will reach a cell, okay. This is suppose your blood vessel. Now listen with all your attention. Suppose this is your, you know, target organ. Clear? So this is your, you know, target cell, okay. Now what happens is this, suppose here is your T4, okay, bound to, you know, thyroid binding globulin, okay. Now what happens is this, with time this affinity increases, the affinity for T4 with respect to TBG decreases, and then what happens is this, not increases, decreases, then what happens is this T4 comes out into the tissue space, okay. Now again here is another, you know, protein. You already know the name of this protein. The name of this transporter is again the same, that is MCT8, okay. Now what happens is this, whenever, you know, T4 comes here, now T4 enters the cell with the help of you know, MCT8 or monocarboxylate, you know, transporter 8. So you know, this is our, you know, tyrosine molecule, right. We need a small revision, okay. So this is our tyrosine molecule, okay. Clear? This is our tyrosine molecule, right. Now a very important point you need to understand is this, that if you want to draw T4, then you know what this particular, you know, tyrosine will have to go through, you know, conjugation and all these things, iodination, then conjugation, right. Then it will look like this, okay. Can you tell me that what is the basic difference between this structure of, you know, this nitrogen and this structure of this nitrogen? Actually this will look like this. This nitrogen will look like this. Can you tell me why? Now listen with all your attention. Chemistry time, okay? Organic chemistry time, sorry, physical chemistry time, whatever. If you know, it's only chemistry time. These divisions only exist for idiots. Now listen. You know what is the hybridization of this nitrogen? What is the hybridization of this nitrogen? sp3, right? But this lone pair is acting like a what? Bond, right? Now listen, as the lone pair of this nitrogen is acting like a bond, therefore it is having what? sp3 hybridization, therefore it is now looking like a tetrahedral structure, okay? Now listen with all your attention. Now we need some bigger space, okay? Now listen. So suppose So suppose this is what? T4, right? This is T4, right? Now a very important point here we need to understand right now is this. Now listen, there are actually, in a cell, cell where, T4 or T3 will enter, that is in a target cell, there are actually enzymes which can convert this T4 into T3, right? Why? Because, T3 is actually, biologically most important form of, what? Our hormone or thyroid hormone. But T4 is the most abundant, right? Now listen, this T3 will have to be converted into what? Sorry, this T4 will have to be converted into T4. Now listen with all your attention. There are three types of deiodinases enzymes, okay? There are three types of deiodinases enzymes. Deiodinases, that means they will remove the iodines. Okay, they will remove iodine. Now what will happen is this. That these deiodinases are of three types: type 1, type 2 and type 3. Type 1, type 2 and type 3. Now listen with all your attention. A very important point here we need to understand before we discuss the deiodinases part. The most important part you need to understand here is this, that for the biological activity of thyroid hormones, you must understand this very carefully. That is, for the biological activity of thyroid hormones, you cannot touch these iodines. For the biological activity of thyroid hormones, you cannot touch these iodines. Why? Because these inner benzene ring iodides or these inner benzene ring iodine molecules are important for the biological activity. Now listen, in reverse T3 you have removed one iodine from here, right? Therefore reverse T3 doesn't have any biological activity. Clear? That means what will majority of the deiodinases will do is they will remove iodine from these outer benzene ring. They will never touch this inner benzene ring. Only idiot deiodinases, one of the best idiot deiodinases is deiodinase 3, type 3. It will actually touch this iodine and it will convert this T4 into reverse T3. But all these two, that means type 1 and type 2 deiodinases. Now listen, type 1 and type 2 deiodinases remove iodine from outer benzene ring. Clear? From outer benzene ring, not from inner one. Clear? Remove iodine from outer benzene ring. They will try to remove what? They will try to remove iodine from outer benzene ring. Clear? It is more or less looking like large intestinal transverse colon. Okay. Now listen. The type 3 deiodinases actually remove iodine from inner benzene ring. Clear? Type 3 deiodinases actually remove iodine from inner benzene ring. Clear? Clear? Now listen with all your attention. Suppose we are now considering a simple reaction in which this T4 molecule is acted upon by, in which this T4 molecule is acted upon by these deiodinases, that is type 1 and type 2. Okay, these deiodinases, type 1 and type 2. Now listen with all your attention. What will happen is this: suppose type 1 or 2. Okay, now a very important part you need to understand here is this, that means this deiodinases will remove iodine from this or this, right? Now listen with all your attention. These deiodinases, all the three deiodinases actually is known by the name of 3', 5' deiodinases. Why? Because they will remove iodine from 3 or 5' carbons, right? 3 or 5, third or fifth carbon. Why? Because in only 3 or fifth carbon, third or fifth carbon, iodines are attached, right? Now listen. So this is the activity of what? Type 1 or type 2, right? So this is T3, right? This is T3, the biologically active, form of what? Biologically active form of thyroid hormones. Okay, this is the biologically active form of thyroid hormones. Okay, now listen with all your attention. Now you must be thinking one thing, that is, what is the basic difference between this type 1 and type 2, right? You must be thinking that what is the difference between this type 1 and type 2, right? Now listen with all your attention. Actually, the difference is in the site, not its working. That means where they are present. Now listen. Actually, type 1 deiodinases are present. Now listen. Type 1 deiodinases are actually present. Type 1 deiodinases are actually present in liver, and all target cells of thyroid hormone. Now listen with all your attention. All the target cells of thyroid hormones, have this type 1 deiodinases. Clear? Now listen with all your attention. It is very, very important for you people to understand is this, that this type 2 is present only in central nervous system and pituitary. Why I am telling pituitary separately, I will talk about that later when we will be talking about the feedback mechanism, hormonal feedback mechanism. That means how T4 and T3 levels in your blood controls the TSH secretion from pituitary. Okay, clear? Okay, so type 1, so what is the basic difference between this type 1 and type 2 deiodinases? Now listen, type 1 deiodinases are present in liver and all other target cells of thyroid hormones. Now what is the difference between this type 1 and type 2? Type 2 is only present in central nervous system and pituitary. Clear? Now then where is your type 3 present, right? Now type 3 is also present in some cells which are the target cells of thyroid hormones. Clear? Now what is the product of this T3? Sorry, what is the product of this type 3 cells? Sorry, type 3 hormones, right? The product of this type 3 hormones, now you already know. Let me sacrifice this part of the board. Now listen. So suppose this is So what is the peculiarity of this type 3 deiodinases? Type 3 deiodinases will actually never touch what? This outer, benzene ring iodines, okay? They will actually touch the more sensitive ones, that is they will remove iodine from this, inner ring, right? Therefore, when any iodine is removed from the inner ring of benzene of thyroid hormones, then that particular thyroid hormone is inactive, okay? That's why this is called reverse T3, okay? It is reverse, this is reverse T3, clear? Okay, so this is reverse T3, okay? Now listen with all your attention. There are at present no biological activity of this reverse T3, okay? But there may be some diagnostic feature about this particular reverse T3, okay, which are used in laboratories nowadays. Okay, so this is the details of deiodinases enzyme, okay? A very important point now we will have to study, that is the role of stress, the role of calorie intake and stress with respect to the activation and deactivation of deiodinases and their role in our basal metabolic rate. Now listen with all your attention. You already know, I guess, by the grace of God, you already know that your thyroid hormones are the master regulator of your BMR or basal metabolic rate, right? Now listen with all your attention. What you will have to know today is this. Now listen. Suppose this is your brain, okay? Okay, this is your brain, and this is your liver, okay? And suppose this is your circulatory system, okay? Suppose this is your circulatory system. Now listen with all your attention, okay? This is very, very important. Now listen. Suppose circulating T4 is way more than circulating T3, right? Now listen. What will happen is this. Some T4 will enter the brain, and some will enter the liver and other tissues like skeletal muscle, smooth muscle, etc. Okay, like skeletal muscle, smooth muscle, etc. Clear? Now here we need to understand a very basic thing, that is, in brain we have type 2 deiodinases, right? And in liver and skeletal muscle, suppose we have type 1 deiodinases. Now listen with all your attention possible on this earth, okay? This type 1 deiodinases are actually inhibited by caloric stress, okay? Why it is important, I will tell you later. Many people are doing, gym nowadays, right? But they don't know this basic physiology. Now listen. Whenever you are taking in less carbohydrate, that means you are developing in your body a caloric stress. And this caloric stress inhibits this type 1 deiodinases, okay? Caloric stress inhibits type 1 deiodinases. Clear? Now listen with all your attention. What you will have to understand now is this, that this caloric stress has no role or has nothing to do with the inhibition of this type 2 deiodinases. That means what? Now listen with all your attention. Many people now, if I tell you the basic thing, if I tell you now, suppose, so if I tell you right now that suppose there are deiodinases, type 1 deiodinases, which are inhibited by caloric stress, suppose. Now what should you think? Suppose if you have a little amount of IQ, suppose you must think or you must tell me now what will happen is this: whenever T4 is going into this organs, then T4 is not converted into T3, right? T4 is not converted into T3 because there is no type 1 deiodinases. Therefore, T4 is not converted into T3, right? Now, a very important point here we need to understand is this: whenever T4 is there in this, liver or any other places, then it will come back to the circulation, right? It will come back to the circulation, right? That means again it will convert to the circulation. Now, an important point you need to understand is this. Now listen, suppose your body is facing a decrease in the amount of T3 concentration. Now, in your in your hypothalamus there is a system, right, which is called feedback inhibition. Now listen, whenever T3 concentration increases, this T3 inhibits the secretion of TSH or thyroid stimulating hormone, right? This T3 actually inhibits the secretion of thyroid stimulating hormone, right? Now listen with all your attention. So, if you don't have T3 in your circulating blood because type 1 deiodinases are inhibited by caloric stress, then what will happen? The concentration of T4 will now increase, right? And therefore, as concentration of T4 is now increased, therefore there will be no T3, and therefore as there is no T3, therefore this inhibition will be removed, right? And therefore TSH concentration will rise, right? TSH concentration will rise. Now listen, many years ago doctors actually thought what you are thinking right now, that this is actually the correct pathway which I have taught. Many years ago doctor thought like that it is actually when T4 cannot be converted into T3 by peripheral tissues, then what will happen? TSH concentration will go up, okay, and that will actually stimulate thyroid glands to produce more and more T4 and T3 and all this. But the actual scenario is this, that whether T4 concentration is high or low, or whether T3 concentration, whenever T4 concentration is high, or whether it is whether T4 concentration is low, it does not have anything to do with the TSH secretion. Now listen with all your attention. T4 concentration directly or indirectly does not have any connection with the TSH secretion. Why Because now listen, we have type 2 deiodinases. What? We have type 2 deiodinases in our brain and in our central nervous system and pituitary. Now what will happen is this type 2 can now convert T4 into T3, and this T3 will actually inhibit. This T3 will directly inhibit this TSH concentration whenever the concentration of T4 goes up. That means what? Whether caloric, now listen with all your attention, whenever you are having a caloric stress, suppose you are eating less amount of carbohydrate, many people do in the name of in the name of dieting, right? But they probably don't know what is dieting. Dieting doesn't mean that you are taking no carbohydrate. It means you are on a keto diet, and keto diet is actually harmful for your body because it will damage your kidneys. Okay, now listen with all your attention. Whenever you are having a calorie deficit diet, then you are having a calorie stress. Calorie stress will now inhibit type 1 deiodinases, but not type 2 deiodinases. That means what will happen? That means in your blood, whenever T4 concentration will rise, this T4 will go to the brain and will be converted into what? This T4 will go to the brain and will get converted into T3 with the help of what? Type 2 deiodinases. Now listen, due to your caloric stress, you have inhibited your own type 1 deiodinases. You have inhibited your own type 1 deiodinases, right? Then what will happen? That means your T4, your peripheral T4 converting tissues will not be able to convert T4 into T3. That means what? That means you will be having a peripheral low T3 concentration. You will be having a peripheral low T3 concentration, and therefore your BMR will decrease and you will feel sick, right? And many gym goers actually prefer this thing as this is having keto flu. Now listen with all your attention, don't talk rubbish. Keto flu has nothing to do with calorie deficit diet. You are having a calorie deficit diet, that means your body is in stress, because one of the best organ which requires carbohydrate a lot, that is your brain. Your brain requires carbohydrate or glucose every millisecond of your life on this earth. Clear? Your brain requires glucose like anything. According to your brain, the best food you can give it is the glucose, not the protein. Clear? Why? Because most of the proteins are mainly polar molecules, and polar molecules cannot pass the blood-brain barrier. Understand this very basic physiology. They cannot pass the blood-brain barrier. Okay? So this is actually the truth of your dieting. Okay? Now, what is the basic thing you need to understand? Take oats in the morning. Why? Because whenever you are starting your day, whenever you are starting your day, you need what? You need some good body balance or something like that, and this will come from T3 and T4. Actually, this will come from T3, and T3 will actually generate metabolic hormones and all these things, and metabolic enzymes. Now listen with all your attention. Take oats or something like that, okay, which is 50% protein and 50% carbohydrate or something like that. Then your caloric stress will be gone, and this inhibition will now be lost, and therefore you can actually go for the rest of the day without having caloric stress. Now, what is the best time to give your body highest amount of protein? In the evening. Why? Because if you give highest amount of protein at night, okay, if you give highest amount of protein at night, now remember this, protein takes way more time, protein and lipid takes way more time than carbohydrate. Now at night you need faster digestion. Because why? Many people don't understand the reason why we take carbohydrate at night. This is because we have to break the fast. Now listen, if you are taking more and more lipids, more and more proteins at night, what will happen is this will stay in your stomach and in your small intestinal large intestine for a long time. And again, if you are taking in the morning some food or something like that, that means what? You are not breaking the fast, you are actually not fasting. How will you lose the extra calorie? To lose extra calorie, take protein on and around 7 p.m. or something like that, and take a low carbohydrate, low protein diet on around 10 p.m. That's it. That's it. Okay? Low carbohydrate diet. Try brown rice. It will, you will enjoy it even more. Okay? Clear? So that is the science of dieting and all these things. Okay? With respect to thyroid hormones. Now we will be talking about in great details how this T3 works in a particular cell. That means we will be talking about the signaling pathways. Okay? All right. So now we will be talking about the receptor signaling pathway of thyroid hormones. Okay? Now before we start that, we will have to talk about in details the structure of thyroid receptors. Now listen with all your attention. You know what is the peculiarity of thyroid receptors? Do you know what is the peculiarity of thyroid receptors? Now listen with all your attention. Now, very important point you need to understand before we talk about thyroid receptor is this. The peculiarity of this thyroid receptor is, they are, suppose this is a cell, okay, these thyroid receptors are nuclear receptors. Thyroid receptors are nuclear receptors. Okay, the first thing you need to understand, that means these receptors are already present in what? Nucleus. Okay, now listen with all your attention. In our body, okay, thyroid receptors are of two types. Okay, there are two types of thyroid receptors which are present. Okay, number one is thyroid receptor alpha, number two is thyroid receptor beta. Now listen with all your attention. Thyroid receptor alpha is present in chromosome number 17, and thyroid receptor beta is present in chromosome number 3. Now listen, actually I should have told that the genes for thyroid receptor alpha and genes for thyroid receptor beta are actually present in chromosome number 17 and 3. Now you know why I have drawn this 17 small and this 3 because of karyotyping. What is karyotyping? Karyotyping is nothing but sequential arrangement of chromosomes, so that the larger ones are placed at the first and the smaller ones are placed later. Okay, this is actually karyotyping. Now listen, chromosome number 1 has the largest size, that's why it is 1. Okay, now 3 is actually bigger than 17, therefore it looks like this. Now listen, so thyroid receptors alpha and beta are actually present in chromosome number 17 and 17 and 3. Okay, now listen with all your attention. So suppose if I tell you, now listen with all your attention possible. Suppose this is a chromosome. Okay, suppose this is a chromosome. Okay, now listen with all your attention. Suppose this is a gene. Okay, now from this gene an mRNA will produce. Okay, from this gene an mRNA will produce, right? And from this mRNA a protein will be translated, right? A protein will be translated. Now listen with all your attention. What you should know right now is this, that suppose this is a chromosome, and from this chromosome mRNA and hence a protein is coming. Now listen, what is the structure of these thyroid receptors? First you will have to know this. Okay, now listen, I am drawing a linear structure first. So this is the structure of a protein right now. Okay, clear? Now listen with all your attention. Suppose this is the N terminal and this is the C terminal. Okay, suppose this is the N terminal and this is the C terminal. First area near the N terminal is called, this area is called trans activation domain. Okay, trans activation okay, this is called trans activation domain. Okay, now what do you mean by trans activation? What do you mean by trans activation? Now listen, trans activation means, suppose this is a protein. Okay, suppose this is the protein. Okay, now listen, if some ligand bind to this receptor, suppose this is the receptor, protein receptor, and if suppose a ligand binds to these receptors, and suppose this is DNA, and if after binding this proteinaceous ligand, this end of this protein receptor now binds with DNA and opens the zip of DNA double helix, so that transcription can occur smoothly. This thing is called transactivation. This is called transactivation. Okay, this is transactivation. This is transactivation. Clear? Now listen with all your attention. So this is actually the transactivation domain. Okay, the next domain here is called DNA binding and dimerization domain. Okay, this is DNA binding domain or dimerization domain. Okay, now the next domain in this protein is called nuclear localization domain. So this is nuclear localization domain, this is called DNA binding and dimerization domain, and this is called transactivation domain. Okay, so up to now we have discussed 3 domains. Okay, this whole last domain is called hormone binding domain. hormone binding domain. Clear? This whole last domain is called hormone binding domain. Okay. Now a very important point you need to understand is this, that this transactivation and DNA binding domain, DNA binding and dimerization domain actually helps in, the transcription process. Okay. But this hormone binding domain is really important. Why? Because any mutation in this particular domain, okay, any mutation in this particular domain is catastrophic, has catastrophic effect with respect to, thyroid hormone, effects in our body. Clear? Okay. So suppose this is our thyroid receptor alpha. Okay. Now, if I ask you a very much biotechnology or bioinformatics question right now, you will have to understand this question first, then you can answer me. Okay. Now listen, if I tell you there are actually how many domains? 1, 2, 3, 4. So out of these 4 domains, can you tell me which is the most conserved of them all? Suppose this is TR alpha, or this is thyroid receptor alpha, and suppose this is, suppose this is TR beta. Okay. Suppose this is thyroid receptor beta. Now, out of these 4 sequences, so this protein, this TR beta or thyroid receptor beta also have these receptors, also have these domains, also have these domains. Okay. Right. Now, my question is, out of these 4 domains, can you please name those domains which are most, conserved? Among these 4, domains in these 2 proteins, the DNA binding and DNA dimerization domain, that means this is the most conserved. Clear? This is the most conserved. This is the most conserved. Okay. In most of the cases, this is the most conserved. If you want to learn that how can you find out this sequence homology of 2 proteins, okay, now there are multiple bioinformatics programs present right now in this world. Okay, don't worry, first understand this. Number 1 is your BLAST. You know this, what is BLAST? There is another thing called PSI BLAST, okay, and many other. Now I will be giving you here DOI of a paper, okay, in the description. You can actually check that paper and you can actually follow the steps which are told there. Now listen, just don't read the paper, you actually read the paper, and by reading that you try to apply it on your own. Okay. Now there is an important and interesting point here you need to understand. Now listen, the best homology occurs between these 2 regions, right? That is DNA binding and dimerization domain. These 2 matches at almost around 47%. Now listen with all your attention. You know what is homology? What is the real meaning of homology? Do you know that? Now listen, homology means these 2 segments have actually not changed during the course of evolution. That means they are so much important, that is any mutation in these areas of the gene have been neutralized, or they do not have any effect on this, on the working of this particular area. Now listen, what is the special property of this DNA binding and dimerization domain? Now listen, suppose this is your DNA, okay, suppose this is your DNA, okay. Now listen with all your attention. Suppose this is, I will be talking only about the DNA binding and dimerization domain. Now listen, suppose this is the N terminal of the protein and this is the C terminal of the protein. Okay. Now listen, now we will be talking about only this small domain. Okay. Now we will be talking about this small domain only. Okay. Now suppose if I draw like this, that this is the domain Now, whenever in a protein, you will see something like this. Whenever in a protein, in any biochemistry book, if you open, whenever in a protein, you will, if you see something like this, in a double, edged line and like this means it's a protein loop. Okay, it's a protein loop. Okay, it's a protein loop. Clear? Now listen, you know what is this? You know what is this drum? Whenever in a protein you see this drum, these represents alpha helix. Okay, this represents alpha helix. Clear? This represents alpha helix. Okay, now listen with all your attention. What happens is this, now we will be zooming into this area. Now listen with all your attention possible. Now we will be zooming into this area. Now we will be zooming into this area. Okay, now we will have to zoom into this area. Now listen, suppose this is the bigger picture of this area. Okay, clear? Now what is happening here is this. So this is your alpha helix, right? This is your what? Alpha helix. And this is what? This is your alpha helix, right? This is your protein loop, right? This is your protein loop. Now listen with all your attention. A very important point you need to understand here is this, that in between this protein loop and this helix, in this valley area, there is an important thing. You know what is that? There is a simple zinc molecule here. There is a zinc molecule here. Okay, there is zinc molecule here. Okay, there is zinc molecule here. Now listen with all your attention possible. Now listen, this zinc is present, whenever a zinc is present in this type of proteins, okay, which are associated with transcription or something like that, this area containing the this area containing the zinc is called zinc finger motif. This is called zinc finger. This is called zinc finger motif. Zinc finger motif. Okay, this is zinc finger motif. Now listen with all your attention. Zinc finger motifs are specialized, now listen with all your attention possible on this earth. Now zinc finger motifs are specialized, areas of proteins which are associated with DNA, okay, like transcription factors and all, okay, which actually help in, unzipping the DNA. Clear? Which actually helps in unzipping the DNA. Now listen, you need to understand one thing, that how this zinc has been placed between this protein loop. Oh, by the way, what do you mean by this protein loop? This protein loop here means beta pleated sheet. Okay, loop of a protein is actually a special type of beta pleated sheet. Okay, this is beta sheet. So that means in zinc finger motif proteins, this zinc ion is present in between this beta sheet or beta protein loop and alpha helix, right? Now listen with all your attention. So again, draw with me so that you can understand, okay? So here is our special and good looking zinc. Okay, here is our special zinc molecule. Okay, this is our zinc molecule. Now listen, now you know what is coordination bonding, right? Now listen, this zinc will now undergo a coordination bond with two histidine molecules and two cysteine molecules. Okay, now listen, so this is what? Cysteine molecule and this is histidine molecule. Okay, clear? So this zinc has undergone, now this zinc has undergone what? A coordination linkage with two cysteine molecules and two histidine molecules, which are present on this protein, which are part of your protein structure. Now listen with all your attention. A very important point here we need to understand is this, that instead of this histidine, there can be two cysteine. That means histidine may undergo coordination bond or coordinate. Zinc can undergo coordination with two cysteine and two histidine or four cysteines. Okay, two of the cysteines are present in the what? What is this? Alpha helix. This is alpha helix, right? Right. This is alpha helix, and this is protein loop, right? Actually it is what? This protein loop is nothing but what? Beta sheet. Okay, beta sheet as protein loop. Okay, clear? Now listen with all your attention. This whole thing, now listen with all your attention possible. So this here is your N terminal and the distant there is your C terminal. Okay, now you need to understand one very basic thing here, that is, in this case what happens is this, that this whole thing now goes deep into the DNA. Now this whole thing goes deep into the DNA, right? Okay, so this whole thing means what? This is what? Zinc finger motif. This zinc finger motif are specialized areas which are present in proteins which bind with DNA. Clear? Now listen. So suppose this is your DNA. Suppose this is your DNA. Now this whole zinc finger motif, will come down and will attack the major groove of DNA. Now listen with all your attention possible. They will attack the zinc finger motifs, helps in unzipping DNA from major groove. Understand this very carefully. Major groove. They will never attack minor grooves. Clear? Helps in unzipping what? Helps in unzipping DNA from major groove. They will attack the major groove. Okay. Now what will happen is this, due to this unzipping of DNA, when this occurs, due to this unzipping of DNA, what will happen is this, a bubble like structure will form, right? A bubble like structure will form, right? And in these areas, RNA polymerase can now enter and start transcription so that this protein can be produced. Clear? Sorry, not this protein, so that other proteins can be produced. What are the proteins produced after the actions of thyroid hormones are over? Sodium potassium pump, potassium channels, and many other proteins. Okay, like glucogenic enzymes are produced after T3 activation of cell. Okay, clear? And these all together will activate basal metabolic rate, or will change the basal metabolic rate properly. Clear? Now we will be talking about how this whole thing actually works. Now this is actually, this is the part of molecular biology we have already discussed in great details. Go and watch that video, that how these zinc finger motifs actually work. Okay, and there are several other motifs, don't worry about that. Okay, there are many motifs which we have already discussed under the heading of nuclear receptors. Okay, now listen. Now suppose this is a cell. Okay, now suppose this is a cell, and suppose here T4 will now enter by the help of a transporter, that is MCT8, right? Now a very important point here we need to understand is this: actually T4 can enter the cell without the help of any transporter. Okay, now with the help of transporter they can also enter, but without the help of transporters also they can also enter. Okay, so we have got our T4 into the cell. Now when it will come into the cell, that means when it will enter the cytoplasm, what will happen is this T4 will be converted into T3. This T4 will be converted into T3 by the help of enzyme, already, deiodinases, right? Deiodinases. Okay, deiodinases. Now deiodinases, there are many types we have already discussed. Okay, now listen with all your attention. When this deiodinases will be active, then T3 will be formed, which is the biological active form of thyroid hormone, right? Which is the biologically active form of thyroid hormones. Now listen with all your attention. Thyroid receptors are present in the what? In the nucleus, right? So suppose this is your nucleus. Okay. Now what will happen is this T3 will enter. This T3 will enter the nucleus as if there is no barrier. Why? Because T3 is electrically neutral, non-polar, and therefore can cross the phospholipid bilayer. Any non-polar molecule can cross the phospholipid bilayer. Example? Can you give one example? Every steroid hormones can cross the phospholipid bilayer without any transporter help. Clear? But every protein needs a transporter because proteins are polar in nature. Clear? Okay. Now listen. When this T3 enters, what we will have to understand is this. So suppose this is our DNA. Suppose this is our DNA. Suppose this is our DNA. Okay. Now listen. I will not be going into the details of thyroid receptors now. Suppose this is thyroid receptor alpha or beta, whatever you think it is. Okay. Now listen. Suppose this is your thyroid receptor alpha. Now listen. Thyroid receptor alpha are predominant during your developmental stages. Okay. But thyroid receptor beta is also present in your developmental stage. But in your adult stage, thyroid receptor alpha and beta are present in almost equal concentration. Okay. So whatever you write, it is right. Okay. Now listen. Suppose so this is what? TR alpha, suppose. Now what will happen is this TR alpha is actually not work alone. This thyroid receptor never works alone. They form a heterodimer. They form a heterodimer and then they work together. Now that means this TR alpha will now bind with another receptor which is called RXR, which is called RXR. Okay. Clear? This is called RXR. Now when TR alpha binds with RXR, this becomes an active receptor, and then it responds when TR alpha is bound to RXR, then and then only TR alpha responds to the binding of then and then only they responds to the binding of T3 with TR alpha. Okay, then what will happen is this. Now what is the structure of a gene. Now listen, if you want to know the really simplistic structure of a gene, you must understand. Suppose this is a gene. Okay, clear? Suppose this is a gene. Now listen. Now listen. Suppose this is a gene. Now listen. Suppose this is a DNA molecule. Okay. Suppose this is a DNA molecule and this is a gene. Okay. Now what is this? This is a gene proper. This is actually the gene. Okay. This is actually the gene. Okay. This is actually the gene. But the gene can be controlled using switch. Every gene can be controlled in your body using a switch, using not a particular switch, but different genes have different switch. That means your genes can be turned on and off. This is the basic mechanism of lacoperon. Okay, this was shown by lacoperon. Now listen, what happens is this gene is actually controlled, suppose, by this switch. These switches are known by the name of promoters. The switches are called promoters. Clear? That means every gene has a switch, on and off switch, and these on and off switches are called promoters. Now a very simplistic answer is this. What is activators or transcription factors? Now listen, you must understand that promoters also have on and off switch. They are called activators. Clear? Now you must understand here that where TR alpha will bind with DNA, that is actually not the gene. Now listen with all your attention possible on this earth. Where TR alpha is binding, where this TR alpha is binding, actually, where this TR alpha is binding, okay, where this TR alpha is binding is not actually the gene. Suppose gene actually starts from here. Genes actually starts from here. Okay. Now, from where the place where the TR alpha will bind with RXR or retinoid X receptor. RXR means retinoid X receptor. Clear? Retinoid X receptor or RXR. Okay. Now listen, where this TR alpha will bind is known by the name of promoter or controlling elements. Okay. Now listen, they are controlling elements. Okay. Now listen, in case of thyroid hormone thyroid hormone activation cells, that means which cells will be activated by thyroid hormones, these controlling elements are called thyroid response elements or TRE. These controlling elements are called and these thyroid response elements are specialized regions of DNA where this double-stranded DNA will now open. This double-stranded DNA will now open, and where the RNA polymerase will be loaded, and from there, if RNA polymerase sticks to the DNA, then if it enters this gene area, then mRNA transcription will start. Okay, and now listen, what are the products, what are the mRNA products of this gene activation? Okay, the first mRNA product of T3 is, the first mRNA product is sodium potassium pump. Second is glucogenic enzymes. What are glucogenic enzymes? Enzymes required for gluconeogenesis and glycogenesis. Okay, and glycolysis even. Okay, another thing is mitochondrial enzymes. Why? Because these enzymes are required for TCA cycle as well as ETS. Mitochondrial enzymes. Okay, another one is beta adrenergic receptors. Now what is beta and alpha adrenergic receptor? We'll talk about that when we will be discussing the details of pharmacology. Beta adrenergic receptors, and then myosin molecules and myosin heavy chain, and other enzyme and proteins are produced. So these are the basic protein products of gene activation by T3. Okay, so these actually are the proteins which help in BMR and all these other things, or metabolic activity of our body. Clear?